It's my pleasure to start off this semester with our visiting scientist. So Cheryl Logan, who will be here for the year, will be our first speaker of the seminar series this semester. She got her bachelor's at UC Berkeley, where she double majored in molecular and cellular biology and integrative biology. She then went on to do her PhD at Stanford, working with George Sommerow. She then went on to Princeton and did a postdoc there before moving back to California and starting her position at CSUMB, where she's been since 2012. So many of you guys know Cheryl. I know probably a bunch of you guys have collaborated with her or are students on, that she works with. But for those of you that don't know her, she is a physiological ecologist. So she studies the physiological mechanisms that animals use to survive in their environments. And she does this on all ranges of level, from the cellular to the whole organism, and she works in a lot of different environments. So she has research programs that are more local, and she's going to be talking about her research in the Galápagos today. Her research, she's been able to get grants and get funding from a lot of different agencies, NSF, Nat Geo, Fulbright, NOAA, and so she's been able to do quite a bit of exciting research. And so with that, we'll introduce Cheryl.
All right, good afternoon, everyone. It's really exciting to be here, not only to tell you about the research that I've been doing over the past several months, but also as the new visiting scientist this year. I'm looking forward to getting to know those of you that I don't know already. And so today what I'm going to be talking about is some work that I did in the Galapagos Islands between the months of January and July of this year, 2019. I went down there as a Fulbright scholar, and my husband was also granted a Fulbright scholarship, so we both went together. We were working on different projects, but we were able to bring our entire family. So we brought our two kids. We have a 4-year-old and a 6-year-old, and so it was not just a scientific adventure for us, it was a personal adventure for us as well. And I'll show you some pictures of our family adventures, but most of the time I'll be focused on the research that I was doing on corals. So corals, you don't exactly think of the Galapagos when you think of corals. It's a very unique place for corals. The temperature in the Galapagos is very different than most equatorial regions. The temperature varies quite a bit, so because of the unique oceanographic conditions there's multiple currents that are bringing both cold and warm water into the Galapagos archipelago, and so we get really big swings in temperature, and there's also upwelling that occurs. So we get acidic waters and low aragonite saturation states in this area, and so it's harder for corals to calcify. And because corals live in a really narrow temperature range, we see fewer corals than we do in other tropical locations. And just from this picture here, this is Darwin's Arch from Darwin Island, which is the northernmost island in the Galapagos. You can see it doesn't necessarily look like a tropical paradise. It looks more like a temperate region. And when you dive underwater, it's very confusing because you're like, am I at the equator or am I in California? It's cold and then it's warm, and you're seeing all different types of animals that live in both areas. So this is what it's like to work there. And I'll orient you a little bit more to the islands themselves. So the Galapagos is located about 1,000 kilometers west of Ecuador. And it's part of Ecuador, but they sort of operate almost as two different countries because it's really quite difficult to live in the Galapagos even if you're Ecuadorian. There are quite a few restrictions on who's allowed to be a resident there. And so for visitors like me, it's even more difficult to obtain a visa to get there. There's 3 major islands where we have human populations. So we were based here on San Cristobal Island at the Galapagos Science Center. And this is the island that has the capital of the Galapagos. And then Santa Cruz Island is probably the one that most of you have heard of. That's where the Charles Darwin Research Foundation is located. And there's about 15 to 20,000 people that live on Santa Cruz Island, and about 6,000 to 8,000 live on San Cristobal. And then there's also a small population here that lives on the southern eastern side of Isabella Island. So even though this island is the biggest island, the population is very small. And then there's also a pretty small population here on the northern part of Floriana. All the other islands are completely uninhabited, and they're tightly regulated in terms of tourism. Most tourism is still via cruises, but there's a burgeoning land-based tourism industry that's occurring there as well. Okay, So as a Fulbright scholar, the goal of the Fulbright program is to increase mutual understanding between countries, which is something that we're in dire need of right now. And I just wanted to give a plug for the Fulbright in general, because I think a lot of times people think of the Fulbright as being a scholarship that's really more focused on the social sciences and humanities. But there are a lot of Fulbright scholarships that are focused on sciences and engineering. And the one that I applied for was, they were looking specifically for folks studying marine sciences. But even for students, there's opportunities for you all to apply for the Fulbright scholarship to get funding to do work in other countries. And the Fulbright doesn't cover research costs, but it does cover your stipend. And if you have dependents, it helps cover some of the costs of them coming there with you. And as a faculty member, you can either apply as a Fulbright scholar to just do research, or you can do a combination of teaching and research, or teaching only. The scholarship that I had was a combination, so I taught a marine ecology course at the Universidad San Francisco de Quito, which has their marine lab on San Cristobal Island, where I was based. And that was a really great opportunity for me to get to know students within the international program that they teach there. And then there's also quite a number of Ecuadorian students that were in that program as well. And then the rest of the time I spent doing the research that I'll talk to you about in the next slide. But here are a few shots of the incredible wildlife that became very commonplace while we were there. So of course the tortoises, the marine iguanas, blue-footed boobies, rays and sharks everywhere. And then at the end of the trip, my family and I were able to go to the Amazon rainforest and spend about a week in a flooded forest there, where we saw anacondas and caiman alligators and tons of monkeys, and it was just an incredible experience for our kids. And then there's a few shots of some of the folks that were involved in the research. Here, there's some familiar faces that you might see. And then up here, we got to meet the US ambassador to Ecuador, who came and visited the lab that we were at, and we were able to give him presentations based on our research while we were there. That's my husband there, who was studying hammerhead shark nurseries. And then our two kids up here, and then this was a trip to a local village when we were in the Amazon. So now I'm gonna jump and talk more about the research that we were doing. My faculty collaborator at Universidad San Francisco de Quito was Margarita Brandt. This is her pictured. She was involved in all aspects of the research, and she also co-taught the marine ecology course with me at USFQ. And she was a PI on the Nat Geo grant that we had to support the work. Dan Barshis is at Old Dominion University. He's a faculty member there, and he was able to come down for part of the trip to help us with the experiments. And then my amazing two research assistants, both students in the Moss Landing program, Caroline Rodriguez and Steve Ryan, were absolutely instrumental in helping to prepare for the trip and also conduct the experiments while we were there. So as many of you know, coral reefs are an extremely important marine ecosystem. They occupy a very small portion of the ocean, but they provide habitat to about 1/3 of marine fishes and 1/4 of marine species. And they obviously are important economically, for tourism, for coastal protection, food security, as well as natural products. But I'm sure that most of you have also heard in the news that coral bleaching has become a major problem worldwide. And coral bleaching is the process whereby there's a dyssymbiosis between the corals and the zooxanthellae, or the algae that live within the corals. So this relationship, the symbiosis, is really important because it provides the corals food and nutrients, and it provides the algae home and protection. And this can be disrupted. It's called bleaching because the algae also provide the corals color. And this happens during times of environmental stress. Most of the bleaching events that we hear about regularly are due to temperature stress, although heat stress, but cold stress can also cause bleaching, and many other environmental factors can lead to bleaching as well. And then if bleaching occurs for long enough, that puts the coral in a state of peril, and it can lead to mortality if the temperatures stay warm for long enough. And then the coral skeletons themselves can be overgrown by algae, and that makes it much harder for new recruits to come in and repopulate. So across the world, we've seen major massive bleaching events in every major ocean basin. These are dots representing different areas of the ocean, but just showing you how widespread these events have been. The first big one was in 1982-83. I'll talk about that more in a minute. And then the next one was in 1997 and 98. These were both associated with El Nino events. And then since then, we've had multiple events, 2010, 2015, 2016, 2017. There's one happening right now in 2019. And models suggest that by the middle of this century that most corals will be gone. These models are based on the bleaching threshold, which is typically about 1 degree above the maximum summertime mean temperature. And we can use what we know about bleaching thresholds to project forward based on what we know about how temperatures are expected to change under global climate change. So we can use the output from global climate models in combination with what we know about bleaching thresholds to make these kinds of predictions. So this is a paper that I led a few years ago, and the dotted line here represents the trajectory for corals globally if they don't have any capacity for adaptation or acclimatization. But we do know that corals do have some capacity to adapt or acclimatize, and so we wanted to understand how much it would change the outlook if we incorporated some of these mechanisms into these kinds of models. And so what we did was we essentially shifted the duration and the window over which corals were adapted to over time. So for example, in the dotted line, that model is based on mean summertime temperatures that have occurred historically over the previous 100 years. But if you shorten that window, and if corals can adapt to the temperature time frame just 20 years before or 10 years before, you can see how that changes the trajectory. So for example, if they could adapt to the temperature range within the previous 60 years, that would buy corals a fair amount of time. And you can see how that can decrease. Now, of course, we don't know how quickly these processes can occur, and we don't know which mechanisms may be acting the most quickly and in which species, but we do know that there's some capacity. And so trying to understand what mechanisms corals might have in their toolbox in order to respond to changes in temperature is quite important for us to be able to predict what is actually going to happen. So corals in the eastern tropical Pacific, as I said, are very different than those in other tropical areas. They tend to be pretty small and discontinuous. They're not very diverse, so there's very few species, especially compared to what you might expect in the Indo-Pacific, and they're less structurally robust. And again, I mentioned this at the beginning, but this is in part due to the dynamic ocean currents that are within this region. There's a lot of upwelling. There's cold water coming from the Antarctic. And so there's both swings that can get quite warm, especially during El Nino years, and then cool during certain parts of the year when there's upwelling occurring. And there's also lower aragonite saturation in those upwelled waters, which just makes it harder for the corals to calcify. And it also makes it harder for them to recover from bleaching and mortality events because it takes more time for them to recalcify. And so this is an image showing some of the major ocean currents. So we have the Peruvian oceanic current that's bringing, the Humboldt current that's bringing cold waters from Antarctic. We have the Cromwell current that is also cold water. So we generally see that sort of the southern islands and the western islands are getting bathed more so by the cold water. And then we have the Panama current that's coming from the north, which is warm water. And so we see that the northern side of these islands tend to see much warmer temperatures than the southern side. So we have dynamic temperature range across the archipelago. And this is highlighted in these two figures as well. So if we look at mean SST, here you can see that on the western side of Isabella, the temperatures can get quite cold. And I mean, I'm talking very cold, like down to 12 degrees Celsius. So it's almost like diving in Monterey, even though you're just south of the equator. And then as you move to the east, it gets a little bit warmer. And then as you move north to the northernmost islands of Darwin and Wolf, it gets quite a bit warmer. So you have this interesting east-west gradient, and then you also have the north-south gradient. And if you look at the variability, you also see that the northern islands have much lower variability compared to the southern islands. So up to 3 degrees change throughout the year here, and it's that same pattern. So more variable, slightly less variable, and then even less variable in the north. These are some temperature records from Academy Bay, which is the southern side of Santa Cruz Island, right here. So it's, these were collected by the Darwin Research Foundation. And these are SSTs from 1965 to about 2012. And what you can see here is the actual temperature record up here on the top. And then we see accumulated thermal stress on the bottom. So the blue is cold stress and the red is heat stress. And so these are differences from the mean temperature that the site would normally experience. And when we see values of greater than 8 over here, that's indicative of a possible bleaching event or a bleaching and mortality event, depending on how much stress there is. So you can see that in the 1982-83 El Nino event, there was a huge amount of heat stress that could lead to bleaching and mortality, and then same thing again here in 1997 and 98. And in that very first big El Nino event, that's when most corals in the Galapagos died. So 97% of corals died in that event, and that was across the entire archipelago. And at that time, we also saw that 16 out of 17 structural reefs disappeared. So we're going from a fully structural reef to, coral communities after this. And the next event, which you saw in the last slide, which was pretty comparable in terms of the amount of heat stress in 1997 and 98. Oh, actually, hold on. I'm going to show you this plot first. So this is a map that Peter Glynn published last year showing what's currently left after the 1982-83 El Nino event. So the green dots are locations where there is currently a coral community. And if the green dot is filled, like this one up here at Wellington Reef, that means that there's a structural reef present. I would argue that there's also a structural reef present at Wolf based on diving there. But the other sites in the south, none of them have any structural reef. These are all really just coral communities that are left. And then in the red, you see these are areas where there was either a structural reef, if the red circle is filled in, or a community. And these are all locations where there's just simply, been no coral recovery at those sites. And then during the 97-98 event, even though there was about the same amount of heat stress, there was far less bleaching than had occurred in the 1982-83 El Nino event, which is a good thing. And so it was proposed at that time that the coral populations might be more heat tolerant due to differential survival after the initial event. But at the same time, there's also been a record of cold stress bleaching events that have occurred during La Nina events. So there's this is kind of setting up the problem here. And this is what led to my research question. So the first question was, are these recovered populations derived from heat-tolerant survivors, or potentially are they new heat-tolerant recruits that have come from other locations? Now, it's almost impossible to answer this question because we don't have any samples from pre-1982-83. And we also just don't really know what the reefs looked like before that. I mean, there's been a few papers by Peter Glynn and his colleagues that were studying reefs here in the 60s and 70s, but we just have very little information about what the reefs really looked like compared to what we know now. And so the second question is really what I'm going to be focused on. Are the recovering populations more heat tolerant than other populations? And is there a trade-off between heat tolerance and cold tolerance? So it could be that some of these populations are more heat tolerant, but are they now also less cold tolerant? Because that could be a problem given the oceanography. And then also, do the recovered populations perhaps have a wider thermal breadth that enables them to survive better because they're more generalists instead of specialists in terms of temperature? And then another question that I won't be getting into today, but what I'm really excited about is looking at the mechanisms that might be underlying thermal tolerance. So in this talk, not all we've done, but what we've done so far is we've basically done a survey of coral thermal tolerance across the entire archipelago, and we've collected samples that we plan to do genetics work with to look at what the mechanisms underlying thermal tolerance might be. And so hopefully maybe next year I can come back and give a seminar and tell you about that. So the experimental plan for the thermal tolerance work was to essentially try to sample corals from all of those green dots in the map, test phenotypic tolerance to both heat and to cold stress, and then eventually to then examine the possible mechanisms that might be underlying this thermal tolerance. So the plan would be to look at changes in gene and protein expression, and then also to look at the type of symbiont that's living within the corals, because we know that that can also help set the thermal tolerance higher or lower depending on the type of algal symbiont that's present. And so the sites that we went to range across the entire archipelago. I think we hit every single green dot except for Marchena here, and we had meant to get to Marchena, but we were on a cruise that went up here to Darwin and Wolf, and we were going to hit Marchena on the way back, but unfortunately the seas were not very good for us that day, and so we had to abandon that plan. But what I'm going to be focusing on today is the 4 of the sites specifically where we got really good data, and they span sort of that thermal gradient that I talked about. So we have the north to south and the east to west gradient. So I'll be talking about Darwin and Wolf, these northernmost islands that have had the most recovery and also have the most robust reefs. Punta Pit over here on the northern side of San Cristobal Island where I was based, and then Isabella Island, there was a site right here, Concha de Perla, where we visited. And so there were 2 target species that we were collecting, and these are both species that were present at all of the different sites that we visited. The first one is Pavona clavis. It's a massive type coral. And the second one was Passalopera species, because we don't know exactly which species we had. It's extremely difficult to morphologically ID Passalopera, and there's also a lot of conflict in the literature as to whether there's just one species of Passalopera across the entire archipelago or whether there's like 12 different species. So we just grouped it all into Passalopera. And we dove at all of the sites, and we collected 8 colonies per species at each site. And within each of the colonies that we collected, we had to break up the corals into smaller pieces that we would then use to place into different thermal environments for the experiments that we ran. And we would oftentimes have like hammerhead sharks swimming above us as we were collecting corals. It was hard to not get distracted at times. Okay, so this is the system that we used for the heat stress experiments. It's called CBASS. It stands for the Coral Bleaching Autonomous Stress System. And it's made up of actually 4 coolers total, so I'm just showing you half of the system right now. There's plexiglass here in the middle to separate it so that we can have 2 different tanks in each one. So we had a total of 8 tanks. There's lights here to keep the corals happy. And then there's a lot of electronics up here, thanks to Steve Ryan. There's a controller and a power box that power the entire system, and this allows us to increase and decrease the temperature. There's a bucket right here with cold water in it, so we can pump in cold water, and then there's heaters inside each of the tanks. And this system was completely built by hand. And it's using parts that are pretty readily available from places like Home Depot. So if you were to go somewhere far away where there's not a lot of resources and you wanted to build something like this, it would be relatively straightforward to build. It's also very portable. So we're able to fit the entire system into 4 50-pound suitcases that we brought with us on the airplane. And this is also a picture of us running the CBASS system on a boat on our way coming back from one of the islands, either Darwin or Wolf. I can't remember where this picture was taken, but we ran all the experiments on the back of the boat. So it's portable. It has relatively low energy requirements. You could run the entire thing off of a generator. And it allows us to do these really rapid field evaluations of coral bleaching, heat stress, or cold stress. And so it's a great system. One disadvantage is that it has lots of moving parts, and when you're moving, those parts can also move and then they can break. But generally, we were able to keep the system going even while we were running it on the boat. But coral bleaching in the wild typically happens as a result of cumulative heat stress occurring over days to weeks. So this test, as I'll show you in a minute, is occurring over the course of like I think, 12 to 16 hours. And so there's a mismatch in terms of like how much we're simulating what's happening in the wild, but I have colleagues that have done other tests and looked at doing this rapid thermal stress experiment versus a longer-term experiment that does more mimic what happens in the wild. And what we see is, although the absolute temperatures may be different between when the actual bleaching occurs, the relative differences between individuals stays the same. So we can still see relative differences in bleaching tolerance between individuals, between species, and among sites using this super fast, rapid method, which is great because a lot of corals are located in places where there's not a lot of resources. And so as I said, there's 8 tanks, and we set it up so that we had 2 replicate tanks per temperature. And we had so essentially we had 2 coral fragments from the same colony in each of the temperatures. And then we did 2 days of experiments at each site. So the first day would be a heat stress experiment where we would either have a control, corals would either be in the control or at 32, 34, or 35.5. And I'll show you what the ramp looked like in just a minute. And then on the second day, we would do the cold stress experiment. So these are coral fragments from the exact same colonies that were exposed to the heat stress experiment. And so in this case, our control temperature was the same, and then we went to 20, 16, and 12 degrees Celsius. This is the profile of the heat ramp. So all the experiments would begin around 3 p.m. So we'd spend the morning collecting corals. And then there would be a 3-hour ramp up from the control temperature here, a hold for 3 hours at whatever the target temperature was, and then a ramp back down to the control. And then this would be a hold that happened overnight, and then we would sample the corals at 8 a.m. the next morning. So we would measure a couple of different things during the experiment. We would test photochemical efficiency. I'll talk about that in a minute. And then at the very end, we would take photos, and we would also take samples and preserve them for downstream genetics analysis. And then the cold stress experiment basically looks the same, except instead of going up, we're going down now, and to these target temperatures here of 12, 16, and 20. And then we did 3 measurements overall. The first one was a visual color check. So we would use this Coral Watch card to record color just visually. And so this was a very rapid assessment. This is a tool that's used by citizen scientists. And so we were kind of interested in understanding how well this color card matches the other measurements that we would take. So it's quick, but it's obviously subjective because it's some person that's trained to do it, and depending on how well they're trained or how much bias they might have, it can be quite subjective. And also, it only captures the final phase of bleaching. So you're only seeing sort of the end point after most of the physiological stress has occurred. And so there's other ways to look at sort of the process that might be happening before you can visually see bleaching. And then we also took photographs. This is kind of the same method as the first one, except we can normalize the photographs to a Kodak color card and make sure that they are comparable across sites and across species under different experiments and different light levels. So this is also quick and cheap, and it's a little bit less subjective than the color card. And then finally, we looked at photochemical efficiency. So we used a PAM fluorometer, and this allowed us to take measurements pretty quickly, but you have to have a fluorometer, which is about a $3,000 device. So it's not as cheap as the other 2 methods. It's less subjective, and it also captures non-visual signs of a disruption between the algal and the coral symbiosis. And this is, the method is called pulse amplitude modulated fluorometry, and as I said, it's pretty fast and cheap, and it's also non-invasive, so you can take multiple measurements over the course of an experiment. And it's based on looking at the efficiency of photosystem 2, which is the light capturing process in photosynthesis. And you can see some of the values down here on a fragment, on 2 different fragments from 2 different species from this paper. And so you can see the darker colored tissue that's not bleached has higher levels than the bleached values over here. And so there is definitely a correlation that you can see between the FVFM values, or the PAM values that we collected, and the color. Although you can also start to see, sometimes it looks like it's a dark, healthy patch, but the fluorometer can tell us perhaps there might be some pre-signs to bleaching that are occurring that you wouldn't be able to see by eye. So the way we are interpreting the PAM data is values between 0.5 and 0.7 are representative of healthy tissue, corals with peak photosynthetic efficiency, and then values of between 0.3 and 0.4 are indicative of corals that are either bleached or stressed, and then values below 0.3 are dead corals. Okay. So this is the first data slide that I'm showing you. This is from San Cristobal Island. And what we're looking at now is the FVFM value. And this is really what I'm going to be focusing on for the results. And temperature over here along the x-axis for heat stress. So on the left, we have Pavona clavis, and on the right, we have Passalapra. And what you can see is that for healthy or happy corals that are around 26 degrees, which was our control temperature, they have the FVFM values that are above 0.5, at least for Pavona clavis, almost 0.55. And then for Passalapra, you see that the baseline is a little bit higher than for the other species. And you can see as temperature begins to increase that you see a drop-off of these values here, and same for the other species. It looks like from this data slide that the Passalapra may be a little bit more tolerant. We see the bigger drop-off here just between the last two temperatures, whereas we see a more gradual drop-off here for Pavona clavis. And then I'll show you what the data looks like for the cold stress, and then I'm going to back up and show you a bigger picture across multiple sites. So this is the cold stress data now, same species and the same orientation. And what you can see is that if you look at the y-axis, you'll see that the values for the heat stress for the warmest temperature are much lower than the temperatures for the coldest, the FVFM values for the coldest temperature. So the cold stress doesn't seem to induce as much of a effect on photochemical efficiency as heat stress does. But what you see a pretty clear drop-off here, though, as we move from 20 to 16 to 12 degrees for both species. And again, we have sort of a different baseline for both species. So from after this slide, I'm going to be showing you normalized values from the starting FVFM value. So we'll just be looking at change in FVFM over temperature. And I did want to show you some of the coral watch data. So this is the color change from the beginning to the end of the experiment for using this method. And then here we have change in photochemical efficiency, the FVFM value. And so you can see that there is a correlation here. I have them separated by species, and it is significant. But it's not, there's still a lot of variability. And we think that this variability is mostly due to just sort of the biased nature of using the color watch card. And so from here on out, I'm just going to be showing you the FVFM values. And we also are in the process of analyzing the red intensity data from our photos, our normalized photographic method, but I don't have the data here to show you that today. Okay, so now we're looking at 4 different sites, the ones that I mentioned before, ranging from the northernmost islands down to the eastern side of Isabella and the northern side of San Cristobal here. We have both species with Pavona on the top and Passalapra on the bottom. And I also forgot to mention earlier that another reason why we chose these 2 different types of corals is that massive types tend to be more temperature tolerant than branching type corals, although we didn't necessarily see that in this data set. So the dotted line here is just a reference line because values that are about 0.1 less than the starting value are indicative of bleaching stress. And so when the values cross over that dotted line, that's a sign that the coral is very stressed. And the first thing you will probably notice from this slide is that the Isabella corals, the ones from here, tend to be the most sensitive to heat stress. And that's perhaps not surprising because they're also coming from the western side, which experiences the coldest temperatures. The other sites tend to look pretty similar to one another. Punta Pitt had a little bit more sensitivity than Darwin and Wolf did in this case, but the differences aren't as great as what we see between Isabella and all the other sites. And now we're looking at the same data, but for cold stress. So overall, the change or the difference in the FVFM value is much less than what we saw for heat stress, so it's not as stressful. So you don't see any cases where the line actually drops below the dotted reference line. And you also see that there's a lot of variability here, but part of that is just because it's a different y-axis scale than in the previous graph that I showed you. And what we see here is that the differences are somewhat hard to see. We do find that the Isabella corals for Passalapra are more cold sensitive than the other sites. And we also see that Punta Pitt Pavona corals are a little bit more cold sensitive than the other sites as well. And so now I'm going to show you a different way to look at the data where it becomes a little more obvious where the differences are. So now I'm showing you box plots just by temperature, so it's much clearer where the differences are between sites. So first we're looking at Passalapra, and again for the heat stress, it's much more obvious that Isabella is much more sensitive than the other sites. And we also see that the Isabella corals are less cold tolerant than the other sites, although the difference is not nearly as extreme as for the heat stress. Now, this is not what we were expecting because we thought that the Isabella corals would be more cold tolerant because they are used to experiencing colder temperatures than all the other sites. So this was definitely surprising, and I'll talk a little bit more later about one hypothesis we have for why that might be. And then now moving on to Pavona clavis. So the first thing we see again here is that the Isabella corals are less heat tolerant than all the other sites. And remember, these are two different species. So it seems like the local thermal history is driving the thermal tolerance in this location. And then for the cold tolerance, we see that Isabella is actually not different from the other sites. The only site that was significantly different was Punta Pitt, showing slightly more sensitivity. And Punta Pitt is on the northern side of San Cristobal, so it does generally experience warmer temperatures, and it doesn't see as cool temperatures as Isabella does. All right. Okay, so this was a study that came out in 2014 that gives us a little more perspective on what might be happening in Isabella for the Passalapra corals. So we saw that they seem to be most sensitive to heat stress and to cold stress. And this paper came out showing that all the corals in this lagoon, which is the same lagoon where we sampled, are actually all clones of one another. So it's a single genotype. And so it seems like either this single genotype was the lone survivor from a bleaching or mortality event in the past, or it could be a lone recruit. We can't really answer that question, but what we do know is that they're basically all genetically identical. And so you would actually think that maybe they would have more similar phenotypes. But if you look at the data, kind of going back to here, there's quite a bit of variability. Like the same amount of variability we see in Isabella is what we see in the other islands. And so, let me go back. Here we go. And so this probably just has to do with phenotypic plasticity. So these corals are living over a range of depths and experiencing different water currents. And so there's different reasons why we might see differences in thermal tolerance among the clones, or there could be somatic mutations that have occurred in this place. But we thought that that was quite interesting that these are an incredibly sensitive population because they're both heat and cold sensitive, and they have no genetic diversity within them. So that's not a good sign for this site. So overall, what we've done at this point is we've done this first assessment of coral thermal tolerance No one had ever looked at coral thermal tolerance in the Galapagos before, and so now we have a map kind of looking at across this wide temperature gradient of what's happening with corals in the Galapagos. And what we see is that the Isabella corals seem to be most heat sensitive compared to all the other sites, and at least for Passalapra, they're also more cold sensitive. We do see that the corals in the northernmost islands of Darwin and Wolf, which are also the most recovered populations, have the widest thermal tolerance. And that is what we expected to see at the beginning because those populations are doing quite well. And they have the ability to withstand both cold and heat stress better than the other sites, at least. We also saw that thermal tolerance is generally more similar by site than it was between the different species. And so we think that this suggests that acclimatization to local conditions might be a driving factor in what's setting the thermal tolerance of these corals. And also, because we saw such variation even within that clonal population, that suggests that there's acclimatization to local conditions occurring too, even within the same genotype. And then finally, just highlighting this idea of high variability in thermal tolerance among clones is a really important aspect, I think, because that tells us that, you know, if there is a bleaching event, there may be, or if there's a heat stress event, there may be differential bleaching, and so there may be differential survival. Whereas if they all were exactly the same and they all got bleached at the same time, then the entire population would be wiped out. And so what are some of the broader implications of this work and next steps? So it seems that the northern Galapagos populations might be more tolerant to future warming to some extent. I mean, with unabated warming, they're not going to survive either. But at least compared to the other sites, they're pretty remote. There's not that many tourists that visit them. There's not that much land-based pollution. They're very far away from where most humans are. So that's good. But we also think that there might need to be better protection and management for the corals on Isabella Island. And we presented this data to the Galapagos National Park while we were there, and they're very interested in sort of understanding what we can do for the Isabella corals. The location where we collected these corals is called Concha de Perla, and it's right near town. It's very accessible for tourists, land-based tourists. You can just walk there like from your hotel or your hostel room. And most people are snorkeling there. The corals are really shallow. It's very easy, and they're also very fragile in that lagoon, so it's very easy to like just kick them and break them and knock them over if you don't know what you're doing. And so the park is kind of trying to think about different ways that they could help protect the corals in this lagoon. And then the amount of phenotypic variability that we see within the clonal population and then just within all the populations in general can be thought of as a good sign. But as I mentioned at the beginning, I'm really interested in sort of what are the mechanisms underlying thermal tolerance and a wider thermal range. And so thinking about what makes a tough individual, what's underlying the thermal tolerance in those individuals is kind of my next step. So right now I've shown you pretty much just population level differences among the sites. But now my next task is to dig in and find those specific individuals that may be more tolerant than others. And then look at the genetics underlying what might be driving that. And so just to take even a broader step back, I wanted to talk about a recent report that I was involved in. I was a co-author on this. It was a committee that was put together by the National Academy of Sciences because corals are, especially in the Caribbean, are not doing well at all. And with these continuing bleaching events, managers there are extremely worried that corals in the Caribbean are just going to be wiped out completely. And so this was an extremely controversial topic like 10 to 15 years ago, but with ongoing bleaching events, now all coral reef scientists are talking about what can we do, what types of human interventions can we implement to intervene, or at least to buy some time for corals as temperatures continue to warm. And so as part of this report, we reviewed 23 different possible human interventions, from everything to translocating corals from places like the Red Sea, where they're really thermally tolerant, into other locations where they might not be as tolerant, or growing corals in the lab that we know to be extremely hardy. So identifying hardy genotypes in the wild, bringing those into coral nurseries, growing them up, and then outplanting them back into their natural environment. There's also a lot of talk about GMO corals, so can we genetically modify corals to increase their thermal tolerance? That's also a very controversial issue because if you increase or select for one particular trait, you're probably losing other traits that might actually be important for those individuals. But there's a lot of work going on right now, especially in Australia, doing these kinds of things. There's also geoengineering approaches, so we can potentially shade areas of the ocean. How scalable those are, you know, there's a lot of talk about whether that's realistic. And there's also talk about microbiome manipulation, so trying to basically increase thermal tolerance by either manipulating the bacterial community within the coral or the zooxanthellae community, the algal symbionts, with algae that are more heat tolerant. And so we reviewed all of these in terms of just what is the state of the science. Like, are people actually doing any of these things in small areas or regions? How scalable are they? How much do they cost? We didn't make any recommendations about what should or shouldn't be done in this report. But the management community is very excited about this report and learning more about these types of interventions. And then we have another report in press as part of the same committee that reviews different decision-making frameworks that could be used by managers to decide whether or not to implement some of these. So in that decision framework, there's different risks and benefits that are associated with each of the different interventions. And so a manager can use this framework in consultation with stakeholders to help develop ideas about whether or not any of these options might be something that they want to implement. If you're a reef manager and you simply don't have any reef left to manage, there is a lot of pressure that we got as committee members from the managers saying, what should we do? We don't want the corals just to be gone. Like, what options do we have? Whereas the scene is a little bit different in Australia, where they're also looking at many of these different human interventions, but the Great Barrier Reef is doing much better than what we have going on in the Caribbean. And so there's a lot of collaboration right now between the managers of the Great Barrier Reef and the managers of the Caribbean, thinking about, like, well, could we implement some of these at a small scale in the Caribbean where things are already really bad, and then learn something about whether or not these are the kinds of interventions we might want to scale up to other regions. So there is some hope, although, you know, many of these interventions have pretty nasty trade-offs if things go wrong and risks associated with them. And so obviously the best thing we can do is to reduce anthropogenic CO2 emissions. So with that, sorry, I just want to thank all of my funding sources and all the amazing people that were involved in this project. So Nat Geo funded all the field work. Fulbright funded my time there and my family's ability to be there with me. The National Park helped us to be able to actually go to all of the different sites. We had to have a park ranger with us whenever we went diving. The Galapagos Science Center is where I was based, and these are the amazing people that run the center there. It's co-run by UNC Chapel Hill and USFQ. Universidad San Francisco de Quito was my host institution, and that's where I taught the marine ecology course through. Of course, CSUMB, and Galakiwi was one of the hotels that we stayed at, and we ran experiments out on one of their balconies. And we became really good friends with the owner of the hotel, so that was great. And then the crew of the vessels that we chartered, and then many of the students and research assistants that helped us out. And then also a big thanks to the marine ecology course students who participated in the stress experiments that were done on San Cristobal Island. And these are two of the students that really helped out after the course and continued working on the project through the rest of the semester. And then Daniel is an undergrad at CSUMB that helped out with some of the data analysis this summer. And this is the amazing crew that came with us to Darwin and Wolf, and, well, it was supposed to be Marchena also, but we didn't make it there. So, yeah, thank you. I'll take any questions.
What you call bleaching occurred, was it because the microalgae left, or was it because they became less pigmented themselves?
That is a good question.
We presume that it's because they left, but we did not do counts, algal counts, so we don't know for sure.
The FV part of the FV over FM, that's FM minus FO, did you lose fluorescence as the?
Yes. Yeah.
Because FV over FM has no dimensions, it's a relative value.
Yeah.
But the actual fluorescence became lower as the bleaching occurred.
Yes.
And then did it go back?
What do you mean, did it go back?
Like when you warm them or you cool them, when you let the temperature.
Oh, when it returned back? That's a good question. So we only did the FVFM measurements at the end of the day after the ramp had finished in the dark because we did dark acclimated measurements. And so we didn't take another measurement in the morning. So I don't know. We never tested that. Yeah, it's good. Good question, yeah.
So you held the temperature up at 35 for 3 hours. Mm-hmm. And that's a pretty steep incline. Would you get the same results, do you think, if you were down at 30 or 31 for a longer time period?
Yes, and I mentioned that there's been studies that have compared this acute sort of rapid thermal stress experiment with longer term, more realistic experiments. And what we tend to see is that the absolute temperature is going to be different. It's going to be lower for the longer term stress because this would probably kill the corals after like another 24 hours. But the relative differences are the same. So if you see a heat tolerant individual in one test, you see that individual also is the most heat tolerant in the longer term cumulative heat stress experiment. So relatively, we see the same differences. Yeah. June.
So given that this area is so protected, I was wondering if you could elaborate on what your process of trying to get permits to do this. And I noticed you were saying that you have an N of 2 per treatment tank, right, per site. So like ideally we wanted to have more.
Well, that's 2 fragments from the same colony.
Oh, got it.
Yeah, so 8 colonies, so there'd be 16 per temperature per site, total, per species, yeah.
So you kind of taking the average, but still,
Yeah.
That's better in my head. But was it really hard to get approval to collect these fragments?
Yes, yeah. So my collaborator, Margarita Brandt at Universidad San Francisco de Quito, was absolutely instrumental in that, and also the Galapagos Science Center and working with the National Park. So she had already applied for permits long before I got down there. And so there were permits that we had to have from the Ecuadorian government. There were permits that we had to have from the Galapagos National Park. And then there were permits that we had to have just to do the experiments themselves, which was different from just collecting them. So there were 3 different types of permits that we had to have, and each time we went out, we had to get approval, like an extra level of approval. So we'd have to walk over to the Galapagos National Park and like sit in their office for like a really long time and wait and hope that they would approve it, and they almost always did. But then you also had to have somebody from the park with you on all the collection trips. So like that's usually a diver, so you had to make sure there was a diver available. And in the end, we didn't have a diver from the park on every single dive that we went on, but we did have them much of the time. Yeah. So and then there's permits, of course, to get the samples out of the country, and I'm not going to talk about that. It's also complicated. Yes.
Did all of the corals have the same type of symbiont within them?
So we don't know yet. So we've collected the samples and we are going to look at that, but we haven't had time to do that yet. All we have right now is the phenotypic data from the field and the samples that are going to be analyzed in the future.
When you go back in a few years, how do you expect ocean acidification to complicate that?
Yes, that's a good question. So, I mean, the corals in this area are already living in really, really low aragonite water, so it's already quite difficult for them to calcify. And so that's one of the reasons why we can kind of use the Galapagos as a case study of like what might be happening in the future in other locations as ocean acidification occurs. But it's definitely going to make it harder for them to calcify. Their structures are already very like loose and compared to other sites. And so with the temperature and ocean acidification happening in tandem, it's not going to be great for them. And what I expect to see is that in the northern islands where there's less upwelling and the corals are already more thermally tolerant, that they're probably going to do okay. But in the southern islands, I mean, these are already really patchy communities. And also speaking to June's question, like, you know, how many fragments could we take? These are small colonies for the most part. So like, if you're taking 16 little fragments from a single colony, you're not destroying the colony, but like you're you know, it's not going to be happy afterwards. And so we did try to limit the amount of fragments that we sampled overall. Yeah.
Is it, you had started off the top with lots of pictures of dead, dying croppers, et cetera. Is there no more croppers?
Actually, I shouldn't have included those photos. I was just looking for bleached corals. There's no cropera in Galapagos. Yeah, the two main species are Passalapra and Pavona, and then as you move north, we also see Porites. And in Darwin and Wolf, like Porites or Pavona is probably the dominant species, probably Porites, but there's very little Porites in the southern islands, and so we wanted to choose species that we knew we could get at all the different sites. Yeah. Yeah. No, it's a different species. Yeah. I don't think we saw any Montastria there, though. Question.
In the Isabella Islands for the Passalapra corals, there seems to be a lot of variability. And what you think in the other sites, the site is the driving factor for the thermal tolerance. What do you think may be driving that variability?
I mean, it's hard to say. We see overall the population, I mean, there's still a big decrease in thermal tolerance compared to the other sites, even though there's still variability among individuals. But that individual variability, like I said, it was really surprising. When we saw the paper and saw that they were clones and we saw that there wasn't any less variability compared to the other sites, I didn't know what to think initially. And I mean, all I can say is that it may have to do with phenotypic plasticity within the site. You know, there's differences in depth, so there's differences in temperature that they're experiencing locally. There's probably some error also associated with our experiments, but yet just to see the same amount of variability was really surprising. Yeah, it's a good question. Steve.
On that same topic, as I recall, that particular collection site at Isabella was physically the most protected from the open water.
Yes.
And I wonder if that changes the amount of temperature swing it sees historically. Might have had some effect.
Yeah, it's definitely warmer because it's shallow and it is protected. And so maybe, you know, we thought that it was going to be more cold tolerant. Maybe part of the reason why it's not is because it is in that protected bay. Although the other species that we sampled was outside of the bay. Pavona was outside of that bay. Although we didn't see as much cold sensitivity in Pavona as we did in Passalapra. I don't think we got any Pavona from Concha de Perla, though. I think we got them all from outside, although we saw a few in there.
So the variability that you saw there also made me wonder which organism is driving the temperature sensitivity. Is it the coral or is it the algae?
We don't know. Yeah, so I mean, that's why we want to look at what algal types are present. From papers that I've read, there seems to be far less algal diversity in the Galapagos in general, and there may actually only be one symbiont type, but there's some controversy over that, and so that's one of the reasons we're also interested in looking at what type is actually present. But if it's the same type, then that leads us to think that it's probably the coral that's driving it, but we'll find out.
So what is the mechanism for the coral expelling the zone? And is it an exfoliation?
That's a good question. So we don't really know, actually. Even though we talk about bleaching a lot and there's a lot of people studying it, we still don't know the exact mechanism. And we think that it might be actually multiple mechanisms that are leading to bleaching. So it could be that the coral is expelling the algae. It could be that the algae is bailing on its own. Or it could be some interaction between the two of them. And it could be that different types of stress induce a different bleaching mechanism as well. So we don't know. Yeah. it's a good thesis.
I have a crazy thought here. I'm thinking back to Emily Schmeltzer's thesis a few years ago. She was studying bacterial communities on corals. And so I was just wondering, do you think that since these sites are so different geographically and there could be a lot of variation in bacteria, could the bacterial communities also be contributing to this heat or cold tolerance?
Most definitely, yeah. There's been more and more recent work on that. I'm not as familiar with the literature, but it would be great to team up with someone who could do that kind of analysis with our samples as well, because we preserved them in such a way that we could look at the entire microbiome. Yeah, another project. Yeah.
Thank you very much. If you have more questions, she's around for
Yeah. Thank you.